Welcome to the Longevity Papers podcast. Imagine finding out that the primary reason a patient's brain is aging isn't actually happening in their skull, it's happening in their colon.
Right, and it's communicating via a sort of chemical remote control, which is just wild to think about.
It really is. And for you, the biotech researcher listening to this, joining us in the trenches of the data, you know the narrative has completely shifted. We are way past the era of hunting for, like, a single magical fountain of youth molecule.
Yeah, that was definitely the old paradigm. The field has matured so much.
Exactly. The new era is all about systemic high-resolution mapping and, highly targeted cellular interventions.
We are looking at a fundamentally different biological resolution now. I mean, the literature from just the past month really cements that shift. We're moving away from generalized pathways toward highly localized mechanistic targeting.
Yeah, and we are covering a remarkably strong 4-week period here. So this is from February 12 to March 15, 2026. We've got 5 major papers that provide concrete mechanistic backing across 3 distinct themes.
Right, so we're looking at immune-mediated rejuvenation, the metabolic regulation of inflammation, and, of course, landmark atlacing.
Let's get right into the macro view of that mapping, starting with the Rockefeller Atlas paper. Because, before we can actually intervene in aging, we need to know exactly what is breaking down.
Exactly. And the Rockefeller Atlas gives us this incredibly high-definition cartography of mammalian aging.
They use single-cell transcriptomics, right?
Yeah, across multiple organs over the entire lifespan of both male and female cohorts. So they mapped aging dynamics at just an unprecedented scale.
And they highlight some pretty significant sex differences in aging trajectories, which is super important.
It is. But what really anchors this paper, I think, is how they pinpoint specific depleted cell types. We aren't just talking about, a functional decline here.
Right. It's not just cells getting tired, they're physically vanishing.
Exactly. Complete cellular vanishing. The depletion mapping is really what sets this apart from previous histological studies.
Take mesenchymal stem cells in the bone marrow, for example, or specific populations of endothelial progenitor cells. The Rockefeller group didn't just say, oh, these tissues lose regenerative capacity.
No, they map the actual physical exhaustion and the absence of these specific progenitor pools.
Because as the microenvironment becomes more fibrotic and inflamed with age, these cells are basically driven into senescence or they just undergo apoptosis.
Right, so you literally lose the architectural foundation of the tissue itself. And that depletion concept pairs incredibly well with the new Stanford proteomics paper.
Oh, the Stanford paper is fantastic. They take that atlacing concept and push it directly into precision medicine.
Yeah, they use plasma proteomics to diagnose exactly which specific cell types and which organs in a patient's body are aging the fastest.
And they do this just by looking at circulating protein signatures in a standard blood draw.
Which is huge.
Yeah.
But to truly appreciate the Stanford paper, we have to kind of look at the broader context of biomarker tools we've seen recently.
Like the OIC-MAGE biomarker tool from Harvard and Brigham and Women's Hospital.
Exactly. Or the Blue Zone epigenetic clocks out of UC Berkeley. OMIC-MAGE integrates metabolomics, epigenetics, and clinical biomarker data to give a really accurate biological age prediction.
And the Blue Zone clocks measure DNA methylation patterns from, exceptionally long-lived human populations to gauge systemic aging rates. They are both massive leaps forward.
They are. They give us a fantastic systemic biological age score. But the Stanford proteomics approach is true novelty.
Right, because it moves from a systemic score to a localized diagnostic.
Exactly. The researchers mapped single-cell RNA sequencing data from various organs directly to the plasma proteome.
So by identifying which organ uniquely produces a specific circulating protein, they can just use the concentration of that protein in the blood to infer the biological age of that exact organ. It's like comparing those older epigenetic clocks to a generic check engine light on your dashboard.
That's a great analogy. It tells you something is wrong systemically, but not where.
Right. But the Stanford approach is like having an OBD2 scanner that tells you exactly which spark plug in cylinder number 4 is misfiring.
Yeah. By isolating brain-specific or liver-specific proteins in the plasma that shift with age, they create an actionable target map.
So it's not just telling you your patient is aging. It's telling you, hey, their renal architecture is failing at a much faster rate compared to their hepatic system.
Which is vital data. But looking at this from the perspective of a biotech researcher building a therapeutic pipeline, synthesizing the Rockefeller Atlas with Stanford's proteomics really forces a model change.
Right. How do we actually translate depleted cell types into actionable therapeutic targets?
Well, if a specific progenitor cell is physically depleted from an aging organ, a traditional systemic anti-aging pill, say like a blanket mTOR inhibitor like rapamycin, that isn't going to bring it back.
It just doesn't have the specificity.
No, the therapeutic target has to become the localized microenvironment. You have to rescue the localized niche.
Because if you want exogenous cell therapies or targeted differentiation factors to take hold, you first have to clear out the toxic inflammatory signals that are specific to that fast-aging organ.
Exactly. And rescuing that cellular niche is impossible if the microenvironment is constantly flooded with senescent inflammatory signals.
Which naturally leads us to our next theme. If Stanford's proteomics acts as our targeting system, we now need a mechanism to execute the cleanup.
The cleanup true.
Exactly. Immune-mediated rejuvenation. Specifically, there is a new paper demonstrating the use of engineered CD8 positive T cells for targeted senolysis.
Clearing out those senescent cells.
Yep.
And the mechanism of recognition here is really vital to understand.
They engineered these CD8 positive T cells with a chimeric antigen receptor, right? A KR.
Yeah, a CAR that recognizes specific surface antigens like UPR. And UPR is highly upregulated almost exclusively on the membranes of senescent cells.
So once the T cell receptor binds to that antigen, it forms an immunological synapse.
Right. And it releases perforin and granzymes, which induces apoptosis in the target cell with just absolute surgical precision.
It's fascinating. But putting this in context against traditional pharmacological approaches, we just saw that dihydromyricetin xenotherapy paper recently published in Nature Communications.
Right, a small molecule approach.
Yeah, dihydromyricetin is a small molecule senolytic. It pushes senescent cells into apoptosis by inhibiting their upregulated anti-apoptotic survival pathways, specifically within the BCL2 family.
Because senescent cells are already heavily damaged and kind of primed for cell death, right?
Exactly. So the small molecule just alters the threshold for apoptosis and nudges them over the edge.
Yeah.
Now, it's an elegant mechanism on paper,
Right.
but I'm going to push back a bit on the feasibility of the CD8 positive T cell approach here.
Okay, let's hear it.
Small molecules like dihydromyricetin are incredibly cheap to manufacture. They scale beautifully in a bioreactor, but we know from the oncology world that autologous CAR T cell therapies routinely cost like $500,000 per patient.
Oh, easily. Because of the sheer complexity of apheresis, ex vivo engineering, and reinfusion.
Right. So what does the actual path to human cell therapy look like for a non-acute chronic condition like aging? I mean, is it even realistic?
That is the big question. The manufacturing bottlenecks for autologous cell therapies are a massive hurdle for longevity applications, no doubt.
It seems almost insurmountable for a general population.
It does. However, if we look strictly at the biology, small molecules often have severe off-target toxicity. Dihydromyricetin and other senolytics interact with very broad apoptotic pathways.
That's true. They aren't perfectly selective.
Exactly. When you deploy a systemic small molecule in an aged, frail patient, it goes everywhere the blood goes. The therapeutic window between clearing senescent cells and causing collateral tissue damage in healthy, stressed cells is often dangerously narrow.
Ah, I see. So the precision of the engineered cell therapy justifies the manufacturing leap.
It has to. It's a necessary model change. The CD8 positive T cell can traffic directly to the inflamed, fast-aging organ that Stanford's proteomics identified, cross the endothelial barrier, and eliminate the senescent cells while leaving the healthy neighboring cells completely untouched.
But cost is still an issue.
Right, which is why the long-term viability relies entirely on the development of allogeneic, off-the-shelf engineered T cells.
Oh, so knocking out the endogenous T cell receptors and MHC class I molecules to evade immune rejection.
Exactly. If we can do that, you can mass produce these senolytic T cells from healthy donors. Mechanistically, T cell-driven senolysis is just vastly superior to pharmacological approaches if you want to avoid off-target toxicity in complex aged microenvironments.
That makes a lot of sense. But those engineered T cells don't operate in a vacuum, though. I mean, you can design the perfect senolytic cell therapy, but if the patient's systemic environment is highly inflammatory...
Those T cells will just face exhaustion and fail.
Right, which shifts our focus to the systemic crosstalk driving that inflammation. Specifically, the microbiome, the brain, and cellular metabolism.
The integration of these systems is where the most actionable insights are emerging right now. A prime example is this new gut-brain axis paper.
the one exploring how specific gut dysbiosis accelerates brain aging. Here's where it gets really interesting. They focus heavily on eotaxin 1, or CCL11.
Right, which is famous in the classic parabiosis literature.
Exactly. Anyone familiar with those old experiments where the circulatory systems of young and old mice were sutured together? Knows eotaxin 1.
It's one of the primary pro-aging factors identified in old blood.
Right, and it actively impairs neurogenesis and cognitive function when you introduce it into a young mouse. It crosses the blood-brain barrier and just suppresses neural stem cell proliferation.
But what is so fascinating about this new paper is that it traces the origin of that circulating eotaxin 1 directly back to the gut.
Right, so what specific bacterial species or metabolites are actually driving this?
Well, it isn't just a single pathogenic bacterial strain causing the issue. It's an age-related dysbiosis that compromises the intestinal epithelial barrier.
Leaky gut.
Exactly, leaky gut. When that barrier becomes permeable, bacterial endotoxins, specifically lipopolysaccharides or LPS, enter the systemic circulation.
And this is where the mechanistic details are just incredible, because the gut bacteria aren't secreting the eotaxin 1 themselves.
No, the circulating LPS triggers peripheral macrophages to initiate an inflammatory cascade.
Furthermore, the researchers identified aberrant short-chain fatty acid profiles produced by this altered microbiome.
Right, there's a distinct decrease in butyrate production.
And that drop in butyrate alters the epigenetic state of host endothelial cells via histone deacylase inhibition, which acts synergistically with the LPS to massively upregulate eotaxin 1 expression.
So the aging gut is essentially remote controlling the aging of the brain through these metabolic and inflammatory signals.
That is wild. The circulating endotoxins and altered metabolites are basically forcing the host's own immune system to produce the factors that halt neurogenesis.
It's a perfect storm. And that concept of metabolic signals driving immune responses leads perfectly into our next paper.
Right, the one detailing the metabolic regulation of inflammation, specifically the interaction between PP phosphoenolpyruvate and the CGAS STING pathway.
Which is a huge deal. For a long time, we viewed metabolism and immunity as parallel but somewhat distinct systems. This paper fundamentally collapses that wall.
Can you break down the CGA STING pathway for the researchers listening, just to set the stage?
Sure. The CGA STING pathway is a primary driver of sterile inflammation, what we often call inflammaging. CGAS is a cytosolic DNA sensor.
So it's looking for DNA where it shouldn't be.
Exactly. When mitochondria become dysfunctional with age, or when genomic instability causes DNA to leak into the cytoplasm, CGAS detects this misplaced DNA.
And then it sounds the alarm.
Right. It synthesizes a messenger molecule called cGAMP, which activates STING, and that triggers a massive type I interferon response. It acts as a cellular alarm system that is just constantly blaring in old tissues.
But what this paper demonstrates is that PPA physically binds to components of the CGS STING pathway and actually dampens its activation.
Yes, specifically, PPA binds to the STING polymer, preventing the conformational change necessary to recruit downstream inflammatory effectors like TBK1.
So PP is acting as a direct metabolic brake on the innate immune system.
Exactly. And when cellular energy metabolism declines with age, intracellular PP levels drop.
The brake is released.
The brake is released and CGS STING goes into pro-inflammatory overdrive. The implications for this are profound.
Because it provides a direct causal mechanism explaining why metabolic decline drives systemic aging and inflammation.
Right. Glycolysis isn't just producing ATP. Its intermediates are actively gating immune sensors.
But as a biotech researcher, the immediate thought is, how do we actually target this? Is PP supplementation physically feasible as an intervention?
Well, we know PP is a highly charged, unstable molecule.
All right, it has a terrible pharmacokinetic profile. You can't just synthesize PP, put it in a capsule, and expect it to survive first-pass metabolism.
Let alone cross the lipid bilayer to reach the cytoplasm of the senescent cell. So no, direct PPP supplementation is biologically unfeasible. It would be entirely degraded before reaching the target.
So this paper is more of an incremental finding then? Or is it a true novel therapeutic avenue?
Oh, it's a brilliant piece of target validation. It's not a formulation guide. The actual therapeutic avenue is the pathway itself. The goal now is to screen for stable, small molecule mimetics of PP, or allosteric modulators of CGS sting that bind to the exact same pocket that PP utilizes.
Keeping that inflammatory brake engaged even as cellular metabolism wanes. That makes total sense.
It's all about metabolic tuning.
Well, we've covered a massive amount of chemical and biological ground here.
Yeah.
From Stanford's plasma proteomics to engineered T cells, endotoxins, and metabolic intermediates like PP.
We really have
But our final paper for this session takes a hard left turn.
the wild card.
Yes. Because what if we bypass chemistry altogether and intervene at the level of physics?
The work by Xiao and colleagues, this is a collaboration across several Chinese institutions and Imperial College London, and it is undoubtedly the most provocative paper in this batch.
They didn't use a drug, they didn't use a cell therapy, they used 34 terahertz mid-infrared light.
Right.
And they reported a 60% median lifespan extension in C. elegans, which is huge. The proposed mechanism relies on non-thermal resonant absorption.
Essentially, 34 terahertz matches the exact resonant frequency of the phosphate groups found in nucleic acids and phospholipids.
But a 60% lifespan extension, that is an extraordinary claim.
It is an extraordinary claim that demands intense scrutiny and independent replication. However, if we look at the broader landscape of early-stage biological models, we're actually seeing a growing appreciation for biophysics and aging.
Like that fascinating early-stage mechanistic paper from Princeton, the one on ribosome aging that was just posted on the bioarchive.
Exactly. That work explored the physical structural degradation of the ribosomal machinery over time.
Demonstrating how ribosomes physically lose their structural integrity, which causes translational stalling and misfolded proteins.
Right. It highlights that aging isn't exclusively a function of chemical signaling cascades. It's also physical entropy at the molecular level.
To visualize this mid-IR light concept, it's almost like an opera singer hitting the perfect pitch to shatter a glass. But instead of destroying it, it's rejuvenating a worm.
Well, think of it more like a tuned mass damper inside a skyscraper.
Oh, okay.
When wind or earthquakes hit a building, the damper absorbs and neutralizes the chaotic energy to keep the structure intact.
Ah, I see. So in this model, the mid-IR light hits the exact resonant frequency of the phosphate bonds in the worm's DNA and cell membranes.
Right. It absorbs and neutralizes the chaotic age-related thermal entropy, physically stabilizing the molecular structure.
And preventing that age-related degradation. The tuned mass damper analogy captures that perfectly.
It does. But as researchers, we must heavily ground this in skepticism.
Right, because C. elegans is a long way from humans.
A massive leap. While the proof of concept that non-thermal resonant energy can alter biological aging trajectories in a simple organism is fascinating. Translating this to a mammal.
The physical constraints alone are daunting. I mean, a nematode is entirely transparent. The mid-IR light can penetrate its entire organismal structure.
Exactly. In a human, 34 terahertz light wouldn't make it past the outer layers of the epidermis. It would be immediately absorbed by the water in our skin.
So the delivery system for a thick, opaque mammal presents a massive engineering hurdle.
Massive. Even if the resonant stabilization mechanism is perfectly validated in independent replication, translating this into a human intervention would require entirely new modalities.
Like what? Fiber optic networks integrated into the vasculature?
Perhaps.
Uh-huh.
Or implantable resonant emitters. It really pushes the boundaries of how we define a therapeutic intervention.
It definitely does. But whether or not we ever see 34 terahertz light therapy in humans, it forces us to think beyond standard pharmacological models.
Which perfectly encapsulates this entire 4-week stretch of literature, really.
It does. We started with Stanford's macro-level proteomics, tracing aging back to specific failing organs, and the Rockefeller Atlas mapping the physical depletion of stem cell pools.
We explored the push for immense precision, using engineered CD8-positive T cells to act as a targeted cleanup crew.
While navigating the complex systemic crosstalk like gut-derived endotoxins and declining PP levels tuning the CGA sting pathway.
All the way down to the resonant frequency of nucleic acids.
Exactly. So I want to leave you with a final thread to pull. If precision proteomics can soon diagnose our fastest aging organs in real time, and if we can engineer allogeneic T cells to act as a highly targeted circulating cleanup crew.
It really changes the whole picture.
It does.
Yeah.
Does the future of longevity biotech look less like a traditional pharmaceutical cure and more like a continuous, highly personalized software update for the human body?
That is definitely something to think about.
It is something to mull over as you head back to the lab. Thank you for joining us for this session and for bringing your curiosity to the table. We will see you next time as we keep exploring the literature.